Thank you for the kind introduction. Yeah, I want to talk about measuring diffusion coefficients of oxygen ion conductive samples in using a relaxation polarization technique, which involves KPFM. And we already heard a lot about KPFM, so I will start actually with my materials, with this, which is cerium dioxide. And ceria actually is a very versatile material after being promoted as material either for solid oxide fuel cell electrolytes or as an electrode constituent in solid oxide fuel cells, because it can have considerable electron conductivity depending on the exact composition, but also oxygen ion conductivity. Recent applications are more and more focusing on the use at temperatures below 300 degrees C. And the material has a high catalytic activity. And some years ago, it was found that apart from oxygen ions, it can also transport protons by a sort of piggybacking mechanism where the protons are also conducted via the oxygen vacancies in the material. And this has led to some new application possibilities. And one of the newest publications, which is actually shown here, is dealing with an implantable glucose fuel cell, which works in the human body, obviously without heating at a room temperature or body temperature. And the interesting thing is that the transport mechanism is relatively well understood at temperatures where the oxygen vacancies are mobile, which means above 400 degrees C. But below this temperature, it becomes a bit more difficult, as the transport properties cannot be modeled with standard defect models anymore. And nevertheless, the interest for applying ceria is very high. So ways have to be found to get information on this material even at low temperatures. And this is where KPFM comes into play. So we already heard some very nice contributions on KPFM. So I do not need to get into detail on this technique, but what we are basically doing is measuring the contact potential difference between metal-coated tip and our sample, and the the contact potential difference is directly related to the Fermi level of the sample, which is in turn dependent on the local defect chemistry of the sample. So we can sort of see the oxygen vacancy concentration of the sample surface. And oxygen vacancies can also be generated or filled when the sample is taken out of equilibrium. So either you change the oxygen partial pressure of the surrounding environment, or you can, for example, apply a potential gradient to the sample. And the idea of coupling polarization experiment using the AFM tip and subsequent KPFM mapping was actually proposed by the group of Fritz Prinz from Stanford. And they did some initial measurements on this, but did not pursue this any further. And as I have been working with various types of ceria-based material, that was an ideal starting point to have a look at a variety of different materials. We have single crystalline materials, nanocrystalline thin films, and also pellets to test whether this strategy can be used more widely, or if it is only applicable to thin films, which was originally proposed. And what we have been doing is that we, for the non-thin film samples, we actually put our samples, single crystals or pellets, into a resin and polish them to mirror, and then put sort of ring counter electrode on top made of silver paste. And then we did our AFM measurements where we first polarized the sample, and then we did subsequent AFM measurements, KPFM measurements more precisely. And as you can see, when you're polarizing the sample with the very, very small AFM tip, which normally has, due to the coating, a diameter of something like 50 nanometers, you get the voltage drop more or less completely in the area around the contact where the tip is. So this is a huge advantage of this technique, because we can actually see the shape of the introduced gradient. And we can see whether it is isotropic, like here in the image above, or if there is a slight anisotropy, like shown here. And we can also have a look at how it does behave at grain boundaries. Does it fade gradually, or do we see two different relaxation processes? All this can be monitored, and we couldn't see this when we would polarize the whole sample and then do a set of measurements. The weakness, obviously, is that it needs a lot of repetition to get meaningful statistics. And obviously, this technique is also time consuming, because you can see that when we polarize, this is the initial image after 60 seconds, and then we take images every 60 seconds. And this sample actually was quite fast, so we see already a strong decay after 600 seconds. But depending on the crystallinity of the samples, this could take hours. So obviously, these experiments are really, really time consuming. We did a variety of measurements using different acceptor-doped cereal materials, and we can see that there is definitely an effect of dopant concentration and also of the grain boundaries within the material. So first, we had a look at a single crystalline material. This means we only look at the bulk. There are no grain boundaries there, so they don't disturb the measurement. And it was very obvious when looking at gadolinium and yttrium doped samples. So gadolinium and yttrium are both trivalent dopants, should have more or less the same effect. And we had a single crystalline material with 10% gadolinium doping or 20% yttrium doping. And what we could see was that the introduced gradient becomes more intense with increasing oxygen vacancy concentration. So the oxygen vacancy concentration is directly dependent on the dopant concentration. And you can see here that that for the yttrium doped sample, the effect becomes much larger. And we were already able to see something with a polarization bias of minus and plus 3 volts, sorry, which was not possible for the 10 mole% doped sample, where we were only able to see something at plus and minus 5 volts.
You have 3 minutes left.
Yeah, okay. We also did some measurements with pure ceria and materials with very low dopant concentration, and there was no effect visible. So this leads us to the conclusion that the effect is directly dependent on the oxygen vacancy concentration. And on the other hand, we could see that the effect of polarization is less intense if grain boundaries exist, because grain boundaries in ceria have a blocking effect for the oxygen ion transport. So when introducing the effect, comparing here a single crystalline material and a nanocrystalline thin film, we can see that for the single crystal, the effect is much larger, and also the relaxation process is much slower, because we think that for the nanocrystalline material, the relaxation process is faster because of charge redistribution, because it's also well known that in ceria the grain boundaries are electron conductive. So we have blocking effect during the introduction of our effect, and then charge redistribution by electron conductive grain boundaries. And an additional very nice thing is that we can actually check our AFM results and compare them to conventional electrochemical experiments, because we can do a fitting of the relaxation process, which gives us the time constant tau. And tau, on the one hand, gives information about the chemical capacitance and chemical resistance of the sample. And we can also use this information to calculate chemical diffusion coefficients. They are obviously also available by other measurement techniques. So we can at least estimate diffusion coefficient from higher temperatures and then compare this to our measurements at room temperature. The other nice thing is, which we showed very recently, is that we can actually assess single grains in a composite material. So what you can see here is a composite material where ceria are the redder parts, and the bluer or greener parts are a electron conductive spinel material. And this composite is used in a membrane reactor for partial oxidation reactions. So it is very nice to know that we can really measure diffusion coefficients on a single grain basis. And obviously, last but not least, we can have a look at the geometry of our polarization effect and compare, for example, the Debye length of our materials. So yeah, I'm already at the end of my presentation. Before I'm completely at the end, I would like to thank my current only group member, who's Christina Schmidt, and also our project collaborators, Dr. Stefan Baumann, Ludmilla Fischer, Dr. Kiran, and Professor Joachim Meyer. And yeah, I'm open to discussion.